This is a Y-Chromosome microdeletion detection kit, designed by our company based on MLPA-NGS technology. This includes the manual, Y microdeletion probes, MLPA buffer, TAC-DNA ligase buffer, PCR mix, TAC-DNA polymerase, forest green forward primers, and red reverse primers. The 96 reaction kit contains 8 forward primers and 12 reverse primers. Each primer carries a distinct index, enabling 96 different primer combinations. This allows for the completion of 96 reactions in a single pooled sequencing run. This time we will test whether these 6 DNA samples, extracted from whole blood, carry the Y microdeletion. Use a marker to label the PCR tubes for easy identification. Next, take 3 microliters of sample from each centrifuge tube for today's experiment. After adding the samples, centrifuge them to settle at the bottom of the tubes. Turn on the PCR instrument. Place the samples inside. And press the tube caps firmly to prevent evaporation. Adjust the PCR instrument program. The DNA in the samples undergoes thorough denaturation under the high temperature conditions of the PCR instrument. While waiting, you can prepare the probe MLPA buffer mixture. Calculating based on adding 0.75 microliters of probe working solution and 0.75 microliters of MLPA buffer solution per sample. 6 samples require for 0.5 microliters of each, to account for potential losses during handling, and ensure redundancy. We will add 5 microliters of each for mixing. Shake 10 times to mix thoroughly. After the PCR program completes, remove the denatured samples. Add 1.5 microliters of mixing solution to each sample and gently pipette to mix thoroughly. After the liquid addition is completed, centrifuge. After centrifugation, the sample is placed in a PCR instrument for probe hybridization. This process takes 14 to 16 hours continuously. Press the tube cap tightly to reduce evaporation. Set the PCR instrument program. It is now 14 minutes past 18 p.m., and we will proceed with the next experiment at 9 o'clock am tomorrow. It is 9 am on the second day, and we start the operation of probe connection. Next, we will prepare the relevant reagents: TAC DNA ligase buffer, and pure water. Because it is necessary to preheat on a PCR instrument, we use PCR tubes to prepare the mixture. Take 98 microliters of pure water. 14 microliters of ligase buffer. (2.1 microliters of DNA ligase). Pipette and blow to mix thoroughly. Next, put the mixture into the running PCR instrument. Currently, the temperature has dropped to 54 degrees Celsius, and the mixture is preheated at 54 degrees Celsius for 5 minutes. After the preheating is completed, add 16 microliters of ligation mixture to each tube of sample and mix well by pipetting. Before adding to the ligation mix, you can quickly take out the PCR tube and check the bottom, to ensure that the sample does not completely evaporate. If you are confident, this step can also be omitted. Press the bottle cap to reduce evaporation. Next, the probe is connected at 54 degrees Celsius for 20 minutes. While waiting, we can prepare the solutions needed for the next PCR amplification experiment. Take out pure water PCR mix, forest green forward primers, and red reverse primers. Next, let's take a 50 microliter PCR system as an example, to prepare the solution required for the PCR reaction. This time, the forward primer we chose to use is F501. Add 126 microliters of pure water to the centrifuge tube in sequence. "70 microliters of PCR mix. (1.4 microliters of Taq DNA polymerase. 7 microliters of forward primer F501. After the probe connection is completed, set the program to 98 degrees Celsius for 5 minutes. Denature at high temperature, inactivating the ligase. After the enzyme is inactivated, take out the PCR tube containing the sample. Next, take out the mixed solution for PCR amplification that we configured earlier. Add 29 microliters of the mixture to each PCR tube and mix well by pipetting. After adding all of them, add 1 microliter of reverse primer to each tube respectively. It is necessary to plan in advance which forward primer and reverse primer to add to each tube. Each sample cannot use exactly the same forward and reverse primers. Otherwise, after pulling the samples cannot be distinguished by the index on the primers. We use R1012 to R106 for this experiment. Add the reverse primers to the samples in sequence. After adding all the ingredients, mix them thoroughly by centrifugation. After the samples are mixed evenly, PCR amplification is performed according to the procedure. Press the PCR tube to reduce evaporation. Adjust the PCR instrument program. The amplified product can then be sent for sequencing. Before sequencing, you need to fill out a sequencing form, and the template of the sequencing form can be downloaded from our official website. The index sequence corresponding to each primer can be found in the Excel file provided by the company. Here are the name of each index, the corresponding sequence, and the reverse complementary sequence of the sequence. It is only necessary to fill in the names of the forward and reverse primers, used by oneself in one-to-one correspondence with the sample serial numbers. The VLOOKUP formula in the cell can automatically bring up the corresponding sequence based on the input index name. It is important to note here that automatically generated sequences should not be manually modified to avoid logical errors. Just fill in the corresponding sample names and index sequences into the sequencing form. This is the sequencing sheet from our partner company. The formats of sequencing sheets vary among different companies, but all of them require filling in the sample numbers and the corresponding index sequences. Many companies require testing after sample pooling. However, as a PCR product, when opening the lid for mixing samples in the laboratory, it is easy to cause aerosol contamination. It is not recommended to mix samples by yourself unless the laboratory conditions are very good. Sending the original PCR products directly, and having the sequencing company perform pooling in a professional environment, is a safer option that better ensures the accuracy of the results. It should be noted that PCR tubes are relatively fragile and prone to damage, so care should be taken to protect them. Takeout usually needs to be transported with ice packs. If transported at room temperature and delivered within 1 or 2 days, it generally will not affect the results. If you have any questions, please contact us.